think this is my last work for the whole conference. Once this one's done, I'm home. Right, so we shift the gear a little bit and back to the ATMP world. So I used to have a co-presenter. I will need to acknowledge Joe's contribution to this presentation. He did give me a lot of information, and he did grant me this permit to share their information of the company and this facility and project as well. So I want to start with a key word, and this couple of days we've been talking about collaboration. Collaborations between different suppliers, between the CDMOs and clients, between the industry and regulators. We had this collaboration earlier and earlier. We used to have only the CMO. Now we have CDMO with the development. And there are also CRDMO with the research as well. So that really makes sure our inventor company focus on their key delivery, which is the therapy themselves. And with the development, you can partner with your CDMO to further develop your process to a clinical scale, to a commercial scale. So this case study is with the company called Methanex. They are one of the CDMOs in Taiwan. So I am presenting on Therapy Hub project and facility. So even within the manufacturing space, it's not only the manufacturers. We actually collaborate with a lot of different organizations. Our supplier, when we focus on our manufacturing, or hopefully not deviation, we will rely on our suppliers to develop some innovative technology, to develop some equipment system for us. So all our consultants, our material suppliers, specifically for ATMP, that's a very special product. A lot of the starting material, for example, your viral actor, it's so critical to your quality. And now you have a supplier doing this whole thing for you. And you will need to really build this trust and partnership between the two organizations. Of course, our engineering partner who designed our facility and who will further build and deliver our facility. So some just very quick project background, why we're building this facility. I've been working with this company for many years, I can't even remember how long. So we went through a monoclonal antibody facility, phase 1, phase 2. We went through the field finish facility. We are now at the cell therapy facility, and they even moved on to the ADC facility. That's why Joe is so busy and he couldn't come. And this is just to tell everyone the facility requirements is not only for commercial manufacturing, or it's not really for commercial manufacturing. We want to use this facility to develop a multi-products in this facility. And we want to bring this product from the development phase to the clinical phase and to the commercial phase. So there needs to be some flexibility built in the system, while we really need to maintain the phase appropriate approach. The project location is somewhere in Taiwan, and just to show they have different offices and facilities in Taiwan as well. Cell therapy, for now, is really the pilot scale. So, now back to the ATMP. I always want to look at this complex problem bit by bit. How do we peel an onion? It's layer by layer. That's why I always present this pyramid or triangle. Pyramid is my favorite, whether it's shape or it's the real pyramid. On the top is always the patient. What our patient needs. That's our quality target product profile. And based on that, maybe our patient needs a cell therapy, a gene therapy, a vaccine, or just a tablet. And we develop a product to address this need. And we will have a series of critical quality attributes. What are all the important or critical attributes you need to achieve this quality of your products? And then down a layer is our manufacturing process. The manufacturing process of this particular product. What critical process parameter we will need to control, we will need to monitor to make sure we achieve the CQAs and QTPPs we require. And then we have this new term, which was brought by I think it's in 2018, the ISPE Singapore Baseline Guide. And we have this new concept, which is critical design elements. And probably you think that we talk about facility, we talk about equipment, but also on top of those, your procedures, your SOP, just if it's more important, it's equally important. Without your procedures, you could have the same equipment, but then they work differently. So with all this, we do have the science behind our product. Now our final objective is to build a facility, right? But we need to understand what's the science behind it, what's the science behind our product. So what's the unique feature of our product? For example, this particular cellular gene therapy product, we will need to collect a certain amount of cells. I can't really tell the dosage information to everyone. So that's also a very unique feature of this type of product. So maybe we're very much used to the dosage of 3 milligram and 20 milliliter, but with this type of therapy, we're talking about how many cells we need to achieve this treatment. And with these unique features of the product, definitely we have some new process and some specific requirement of this new manufacturing facilities. And now comes the question, what are the new risks associated with the product, with your manufacturing process, and with your manufacturing facility? This is the thing our regulatory requirements will also need to address. So, well, actually, I always print this little pyramid and bring it with me just to remind myself, everything, you look at the complex issue, right? You're always looking at the top, what the patient needs, and the science, and then your process. If you need a copy, look for me after this presentation. So, with all this requirement, a new product, a new therapy, I know we talked about ATMP for ages, and there's so many different presentations, so much information, but still, ATMP is a very, very new area. If anyone telling me, I'm so experienced with it, and I know exactly what to do, I would give a question mark to this person. Every of us are still trying, even our regulators are still trying with this new therapy and the new risks associated. So, but we do still have loads of good references at hand. So, when I want to decide what a company needs to do, what policies, what procedures I need to put in place, what do I look at? The first layer, definitely on top, is the law and regulatory guidance. Now I feel a little shy talking about the regulations in front of regulators and the regulatory aspect, but I'll just try, correct me if I give any wrong information. So from the law level, there's a slight difference between EMA and FDA. So we're all very familiar with the EU GMP. Chapter 1 is your quality system, Chapter 2 is your personnel, so on and so forth. But actually, they are not a law level in the EU region. The law level in the EU region, there are the directives and regulations. So there are also difference between directives and regulations. The regulations are more stringent. On every of the competent authority or country, you will need to follow exactly what's written in the regulation. Well, directives will give you a direction. This is what we require, and then you will need to specify all these very specific requirements for your nation. So the GMP is actually not in those directives and regulations. Here I give you two examples. One is the Directive 2001/83, that's on the human medicine. And the other one, the one below 1394/2007, that's specifically on ATMP. So I give these two examples just to demonstrate ATMP is new. We have our very specific regulation for it, but it's also human medicine. Whatever is required for human medicine, it's still applicable for our ATMP. So it's not entirely a new area. what you very used to in the past may still be applicable for this new area. So don't be scared with these new terms. But for FDA, it's slightly different. You have the CFR, Code of Federal Regulation, and they will have this GMP, we're very much familiar with the Part 2 2011. So that's the GMP written in the law in America. So that's the difference actually between EU and FDA law system. That's why they need to be written slightly different. But they actually convey very, very much similar information. So I've mentioned the EU GMP is actually a regulatory guidance, which is very much we're familiar with, the usual Lex 1 The main part is the Part 1. We have all the annexes supporting the main Part 1. Annex 1 is the hot topic recently, and Annex 2 being our biologics product. And for our ATMP is actually a new part. So we made ATMP an entire Part 4 as the GMP for ATMP product. That actually now differs from the PICS GMP. Before this one, UGMP and PICS GMP, they're equivalent. But after this one, when PICS decided to put this ATMP GMP as Annex 2A, while the rest of the biologics remains an Annex 2B, these two are slightly different. So just talking about the regulatory guidance from FDA perspective, we have this good tissue practice where everyone's very familiar with the process validation guidelines for the industry, and that's for the FDA region. So the new GMP, Usual Lex 4 Part 4, actually gives us a lot of good information on the facility design. When we have a new ATMP facility, what will we need to take care of and pay more attention to? There's a whole new chapter 2 about the risk. Because of this new product is so new, we don't have the entire good understanding or in-depth understanding of the product and process. We will really need to thoroughly assess the risks associated with all these aspects. So I give some examples here. For example, the first one, we mentioned the cross-contamination. So we could have multiple BSCs, the biosafety cabinets, in one clean room. But there needs to be distance between your different BSCs. You can't put them directly side by side or next to each other. Otherwise, you will have this potential risks of cross-contamination. And there are also some really good practices in the industry. If you have two BSCs in the same room, if they are in parallel or on the same line, what's the distance you need to put in between, or they back-to-back, what distance you need to maintain. And this is not to disturb the airflow or not to disturb your operator's activities in two different BSCs. So the other one I give as an example here, the contaminations for the open system. So if we have the production in an open system, that's very, very common for an ATMP product. And it needs to be in a BSC grade A environment, not necessarily BSC, but grade A environment, under the grade B background. Because this one, on the product, I fought so hard to keep the grade B background. The original planning was to have a grade A and a grade C background. That was not entirely aligned with the GMP requirements. So under what circumstances that we can actually have a grade A and a grade C background if that solution can be sterile filtered after you prepare the solution. So GMP is actually giving you very clear requirement what you need to do with your facility. And then the last one, we're talking about contamination with the closed system. A lot of people say, oh great, we just put an isolator in the room, that solves the problem. You have contained environment, doesn't matter you contain contamination to the environment or you just protect your products in this contained environment. But moving the materials in and out of this contained environment, say isolator, could be an issue we need to resolve. Right? If you bring a material into the isolator without a proper sanitization, you actually contaminate your isolator. So a lot of equipment, small equipment we use for ATMP, for example, your G-Rex. It's very difficult to sanitize your G-Rex because the permeable feature on the equipment. So we really need to think thoroughly about all the solutions. It's not I have an isolator that solves all the problem. Otherwise, all the ATMP facility design would be so easy. Talking about the risk assessment on this project, we really had very intensive risk assessment sessions. So firstly, we look at the process quality risk assessment. We look at each step of the manufacturing process. I give three examples here. First one is the cell activation. Each of the steps will need to be looking at what's the critical quality attribute or critical aspect of the step. For example, the cell activation, we're looking at cell viability, the cell count, and then what intervention we have with this step. We may be taking a sample, and that sampling activity could be your source of contamination. Moving on, we could have the transduction. We have the same CQA. We're probably thinking of what's the efficiency of my transduction, what material we use. Now we have the viral vector. How risky is my viral vector for this step, and how I prevent this viral vector to contaminate or cross-contaminate other material. And maybe the next one is the cell depletion. Here, definitely the separation of different cells. I could be using a very advanced system, Clinimax, so to solve some issues and the open manufacturing manipulation issues I have with that process. So on top of the process quality risk assessment, we also did very intensive system closure assessment. So we look at the process flow and our material preparation. For example, I prepared the buffer and also the sampling activities. And all those, am I really using the real functionally closed system? I give a picture here, it's a lure lock. Do you think lure lock is a closed system? If you say yes, think twice. That's my hint. And so professional guidance, the other reference we could actually refer to is, I'll give you an example here. We have ISPE ATMP. This one is on the autologous. I was told there will be new allogeneic guidance coming up, and also the ATMP equipment guidance could be coming up as well. There's some examples, very practical examples and inspirations given from our guideline document. For example, I just take this one, the figure 4.5. If potentially we have a facility for multi-patients, right? Between different patients' material, there could be really severe contamination. So how can we plan all this manufacturing or operation activity, or how do we segregate between the two different operations? We have multimodality is always the hot topic recently, and I have different modalities in my facility, or even I have different patient materials in our facility. This is a reference I took from the PE magazine, from Civil Engineer magazine, which is a very good source for references. Our colleagues from different companies, they publish very good information in this magazine. For example, this one. So if we're looking at the contamination risk just from a microbial risk perspective, we have this low, medium, high, and very high risk. What we're very familiar with for a monoclonal antibody, it's actually very low risk because it's using a non-human cell. So the virus it could contain in that cell is very difficult to pass it on to the human body. So the second group and medium group, right, we talk about allogeneic cell therapy. Well, from a raw material perspective, it's slightly lower risk compared to autologous because it's not from the patient. And we screen our healthy donor to collect their tissue with their blood. So viral vector, everyone we're talking about viral vector, you'd be thinking that's so risky, so dangerous. But a lot of the viral vectors are actually replication incompetent because it means it doesn't replicate themselves. So the risk level is much reduced with this. It's not a natural virus. It's the layman term is man-made virus from our manufacturing facility. So the risk may not be as high as you expected. So now we talk about the facility layout. Before I use AutoCAD to draw anything or 3D, we would definitely want to sketch on the paper first. We discuss what's the function bubbles and all the function rooms you need for this product and process, right? Firstly, I maybe think I need one grade B or grade C process suite, and maybe if this first one is grade C, the second one, I could have a grade B to handle a very open manipulation, and then I probably have multiple of them because I have multimodality in this facility where I have different process steps. And then in between, I could put a commonly shared solution preparation, because the solution preparation just, for this particular facility is not really a therapy dependent. It doesn't really have the cross-contamination issues. So the other thing I can have in common is the formulation and filling room. And then, of course, after that, I have the visual inspection and control rate freezer room. So around the whole, all these functional rooms, I still need to have my corridors, and also very importantly for ATMP products, we have the kitting area where you need to prepare your material, your single-use material in this room, and that space is often neglected and ignored, actually. So what's the reality? I'm running out of time, so I just show you the reality. So ideally, I have a greenfield project. I can do whatever I want, where I need. But in the reality, this type of facility is always a brownfield project. We need to fit out within an existing space. So I have a lot of limitation. There's a column here, and the room shape is not really perfectly square. Now we try to fit it out within this given space of a rented lab area. So we have solution preparation. This one is currently grade C, but we will have some very small volume solution preparation under the grade A with B background in this room, because of a lot of media components, your cellophane, etc. is too small volume to be in a closed process equipment. And then we have a potential expansion area as the grade C area for the future, and then we have really big grade C area for filling and other larger scale of bioreactors in this room. And definitely we have here shown at the grade zone is actually the supporting area with our nitrogen tanks and with our keeping area and our cryopreservation room. So I was just reminded my time is up. So if you have any questions, we can discuss after the session, or just email any of us. Thank you.
Thanks, Shanshan. So in the interest of time, we have to move on. Thanks, Shanshan, for the wonderful presentation. To take note of the contact details so that you can always